Good evening, ladies and gentlemen. Good evening and hello. Welcome to the award ceremony of the 2023 Eppendorf Science Prize for Neurobiology. It's wonderful to see a great crowd of you here today. And I would like to extend a warm welcome to Science, our friends at Science, Eppendorf colleagues, members of the press, and all of those involved in scientific discoveries. I would especially like to welcome members of the prize selection committee and the 18 former and current awardees of the Eppendorf Science Prize. And I would like to welcome also those people who are watching us online. You will probably, some of us will have noticed that one person is missing today, and that is Axel Jans. That's my colleague Axel Jans. He hasn't missed an award ceremony in 22 years, but unfortunately he caught COVID and so wasn't able to travel. So hello, Axel. My name is Carolyn Talbot, and I have been organising and involved in the promotion of the prize for many years. So it's an honour for me to kick off the event this evening. When this Society for Neuroscience convention is in Washington, it's become a tradition that the prize ceremony is held at the AAAS Science headquarters. And I'd like to take this opportunity to thank Zudip and our friends at Science for hosting this event. The last time we were here was in 2017, and Flavio Donato won that year, and I know he's here, I've seen him. Yeah, there he is. Hi, Flavio. As a Brit living in Hamburg, Germany, I don't really know what it's like to live and work in Washington. But have any of you watched the Netflix series House of Cards? Yes, well, admit to it. I'm now on season 5, and I'm astounded every time by the wheelings and dealings of Claire and Frank Underwood as they strive to become president and remain president of the United States. And I think to myself, that can't be right. It can't be true to life, surely. It's so far-fetched. And then, in Germany, we've had the coverage of the election of the Speaker to the House of Representatives. And then also the interventions of Donald Trump. So perhaps House of Cards isn't quite as far-fetched as I thought originally. But let's get back to the reason why we're here this evening. We're here to celebrate the accomplishments of 3 young researchers, Michael Skinnader, Mattia Aimé, and Marissa Scavuzzo. So first up today is Sudip Parikh from AAAS Science. And then Dennis Barger from Eppendorf, North America, president of Eppendorf, North America, will take the stage. He will then introduce Anne Kennedy, who is actually our 2022 winner, but has come today to pass on the baton to Marissa. We will then hear the prize ceremony, the prize presentation itself, in which Peter Stern, senior editor at Science and also prize jury chair, will honour our 2023 awardees. Then we will hear from Michael and Mattia, followed by Marissa. They will tell us more about their award-winning research. I know that sounds a lot of people. Don't worry, you're not going to be here for 2 hours. So, the event is followed by great food. I think you've tasted that already from downstairs. And I would just like to invite you to stick around. So, Zudip, would you like to take the microphone? Zudip is Chief Executive Officer of the American Association for the Advancement of Science and Executive Publisher of the Science family of journals. Thank you.
Good evening, everybody. I hope you're all doing well. Thank you, Carolyn. That's funny, I often have to bring an orientation together for our science and technology policy fellows here at the AAAS. And the first line that I give them is, how many of y'all have seen The West Wing? And some of them raise their hand, and I say, it ain't nothing like that here in Washington. And then I say, how many of you have watched The Americans? And then more hands go up because it's newer. And I say, well, it's not a lot like that. And then I'll say, have you watched The Diplomat? And they'll say, well, yeah, they've watched it. And I'll say, it's not like that. And then I'll say, have you watched House of Cards? And then every hand goes up. And I say, well, it ain't like that either. But you're right, Carolyn. The more and more that you see of this covered worldwide, you realize that it ain't like that because it's worse. Sometimes it's worse. I'll give you a talk for another day. I think we'll be okay, but sometimes it is worse. All right, my job here is to welcome you all to the AAAS. Welcome to the American Association for the Advancement of Science. Welcome to this monument to science engineering here on New York Avenue in Washington, D.C. Thank you so much for being here. I want to thank our partners and collaborators at Eppendorf. This is a long-time partnership, an extraordinary partnership of 22 years that has always served from the very beginning to have the vision of recognizing scientific excellence amongst early career scientists. And I'm going to come back to that here in a second, because when I welcome you to this building's like 25 years old now, but the organization is 175 years old. It's 175 years old, and it was pulled together in 1848 by 87 white men in Philadelphia. And since that time, they were the naturalists, and they were the geologists and the precursors, meteorologists of their day. And they're relatively progressive for their day. But they never imagined an organization that represents the scientific enterprise, where the board of directors is majority women, where the board looks like America, and where their CEO is a guy named Sudhit Parikh. And so in this enterprise, I want you to have a little bit of optimism. So forget about the House of Cards for a second, and remember that organizations, institutions like this can change over time, And now, in our 175th year, we've rededicated ourselves to what are the things that we do as an organization? What are the things that we do? And what we realized is that our job here at the AAAS and at Science and the family of journals is to actually create the scientific community that we think we should have. And so the very first lines of our vision are a boldly inclusive, mobilized, and global scientific community. Each of those three adjectives have a lot of meaning for us, a huge amount of meaning. Inclusive, mobilized, and global scientific community. And if we can create that scientific community, all the wonderful stuff that can come after, all the betterment of humanity can come after that, as implications of having that scientific community. And to get there, you can't just have optimism and a hope, you have to have a plan. And so we have a plan. We have 4 things that we do, 4 things and only 4 things. And tonight I'm going to talk about the first 2 because that's what this prize is about. The first 2 are, the first is recognizing scientific excellence, rewarding scientific excellence and achievement, and ensuring that the future is trained for scientific excellence. And this award fits so neatly into that. It fits completely into that. And it's something that our very first president would have recognized in 1848, and it continues to this day. It is recognizing scientific excellence is going to lead to something bigger, something better, something more important for the world. The second part of our plan is to foster the talent that's required for that. Because we believe that a diversity of thought, a diversity of experience, and a diversity of identity is required if you're going to achieve scientific excellence. It turns out that you have to have a wide aperture to understand the world. And so those first 2 pillars of what we're trying to accomplish is in deep partnership with Eppendorf. Because Eppendorf isn't quite 175 years old, but they're 78 years old, I think, or something close to that. And it's an organization that recognizes two of those basic tenets. One is scientific excellence and discovery. The fact that basic research is so fundamental, so important to the future, that some of the things that we're doing in basic research, we don't know exactly what they're going to lead to, but they're going to lead to something. Michael Faraday, when he was asked, what's so good about this electricity stuff? He said, I don't know, but someday you're going to tax it. And I don't know about all the neurobiology that you all are doing, but someday we're going to tax it. I know that, thanks to House of Cards. And so that is right in front of Eppendorf and science and the AAAS. The second piece is about early career scientists, recognizing that there are many things that are not going so well in the scientific enterprise. One of those is that the average age of the first R01 that's received, the average age a scientist is when they receive their first R01 grant is 42. 42. And that's challenging in a world where so much discovery and innovation is happening in our 20s and 30s. And so Eppendorf and science, we recognize that we've got to create recognitions and awards so that can move earlier. So that in the process of the scientific enterprise can move earlier in people's careers. And so you'll see that there's a common theme in every award that science gives. And it's very much about scientific achievement and about early career scientists. And that's why we're so proud to have Eppendorf as a partner. Now, the last thing I'll tell you up here is that I believe that we live in a golden era of science. 4 years ago when I took this job, I still dreamt about the possibility of having lunch with Einstein or breakfast with Newton in 1620. I thought, gosh, what would it be like to live in one of the golden eras of science? Or even in the 60s when we worked out the central dogma of biology. But it turns out, after 4 years in this job, I realize that this is the golden era of science. This moment. We know more about the universe, we know more about this planet, and we know more about ourselves than we ever have before. And the pace of that discovery is only quickened. Now, there are challenges. There are many challenges, and there are many problems, and some of them are tied directly to what Carolyn started us off on. But my belief and my optimism stems from the work of scientists like we're going to hear from tonight. Because I am fully confident that that progress is going to continue, and that it's up to the rest of us to ensure that they have the support, and they have the funding, and they have the freedom to discover what comes next. So I'm so excited to be here. Welcome to AAAS. Welcome to our award winners. And now I'm going to turn it over to Dennis Barger from Eppendorf, our true partner.
Great, thank you. Thank you, Sidip, and thank you, Science AAAS, for hosting us. So yeah, the first thing that Caroline would mention is that I'm not Axel. So you're expecting to see Axel. I usually run Caroline's part in the beginning. Axel has the more formal part of the message from Eppendorf. So my part is usually a little bit more ad lib, and theirs is a little bit more constructed. So I'm going to try, because I know Axel's online, so I'm going to try to stay to the script. The fact that I began to modify the script, right, and we began to talk about it. I think I received it 3 or 4 times in an email. Various formats, the last one, PDF password protected. So I'm guessing, Axel, that I won't try to go too far from your script. And I appreciate all the effort you put into this, and I do have it. It's right here. All right. So it's a challenge for me personally to stay on script. So yes, good evening and welcome. On behalf of Eppendorf Worldwide, we'd like to welcome you to this year's prize ceremony. It's an honor and a pleasure to be with you this evening. Firstly, I'd like to thank Sudip and our Science AAAS for their generous hospitality in hosting this event and partnering with us here in Washington, D.C. and working together with us over 22 years, as you heard, on this prestigious award. We look forward to continuing this partnership for many years to come. And so, can you believe it? It's been 22 years. The prize has been awarded 22 times since its inception in 2002. We have over 70 winners and finalists that are represented by that body of work, many of whom have gone on to be leading scientists in their field. All the awardees have had the chance to come together once a year and share their science in their own alumni meetings, which happened yesterday. So yesterday, I think we had about 20 members here for an alumni meeting in which they could both share offline their updates, and 3 of them actually presented work that they presented their work from the more contemporaries. It was really interesting to see what they were up to at this time. So I'd like to congratulate our 3 finalists this year. So Marissa Scavuzzo, Michael Schneider, and Mattia Emi on their fantastic success in their work and in being selected as this year's awardees from a large pool of outstanding applicants. We look forward to hearing more about your work tonight. As you saw in the program, you're going to hear from them later on. Many of you are wondering how we find these talented researchers, and it is one of the special things about the Eppendorf Science Prize. So young researchers can apply by themselves, right? The application process is not complicated, so the potential winners really hold their fate in terms of being selected in their own hands. Earlier this year, we caught up with Tiago, and I think he was here yesterday morning as well, I think he's in the back, who had said that when he had already decided, so he was our prize winner in 2011, and he had already decided not to apply because he thought he didn't have a chance of winning. And like all good scientists, he changed his mind over a beer with a colleague, so who brought up the topic and encouraged him to go for it. And as Tiago says, this highlights not only the importance of being surrounded by supportive colleagues, but perhaps the value of sharing a beer or two. There aren't many prizes that you can apply for by yourself, and specifically which are aimed at young researchers. So I just sort of second the sentiments that you heard earlier about the importance of that to both Eppendorf and Science. The Eppendorf Science Prize for Neurobiology specifically seeks out young scientists at the start of their careers. The prize spotlights exceptional scientists who, in a very short space of time, have made fantastic scientific discoveries. Perhaps not long after receiving their PhD, are still 35 years of age or younger, who are postdocs or often just setting up their own labs. These hidden gems not only do great science, but they can also write about it in a 1,000-word essay in a compelling and articulate manner and communicate to the broader audience why they spend so many of their waking hours in their lab. And that's the condition of the prize. When you submit it, it's a 1,000-word essay, and you really have to catch the reader's attention at the beginning. In a tough financial climate, when there are no automatic grant-generating machines out there, getting noticed is often half the battle. So we hope that the visibility that the Eppendorf Science Prize can provide or play may be a crucial role in your downstream success for that. So many thanks go to the prize committee for doing such an excellent job in selecting this year's winners and finalists. So Rosa Cossart from the Mediterranean Institute of Neurobiology in France, Virginia Lee from the University of Pennsylvania, Amos Matsiko from Science AAAS, and Shigeki Watanabe from Johns Hopkins University School of Medicine. Shigeki was our grand prize winner in 2015, and it's great to see, once again, a past winner as now a member of the prize committee. And I think he may be here this evening. Right back there. Thank you for your participation. So with that, of course, a big thank you goes to Peter Stern, Senior Editor at Science, for once again heading the prize committee. Peter, I know this is a lot of work, and we say this every year, but your enthusiasm for the prize and its awardees is amazing. Under your leadership, the jury never fails to select the best scientists to win this award. The quality of the prize is dependent on the jury. The high standard of their work makes sure that only the best and correct winners are chosen. And it's important to note here that Eppendorf does not participate as part of the selection. That's Dr. Stern and his committee. From an Eppendorf perspective, the Eppendorf and Science Prize for Neurobiology goes a long way back. It embodies the vision of our two founders, Dr. Heinrich Nettler and Dr. Hans Hinz, who in 1945 were eager to improve people's living conditions. Their vision remains the heart at what Eppendorf is today. By honoring young scientists, the Eppendorf and Science Prize is our way of empowering scientists to shape the world into a more livable place. It's wonderful that we can come together this evening and celebrate these young researchers and their trailblazing achievements. Before they take the stage, it's my pleasure to welcome back Ann Kennedy, who was the grand prize winner from last year. Ann is assistant professor at Northwestern University, Chicago, won the 2022 prize for her work on aggression. By comparing the activity of neurons in multiple deep brain regions, she identifies differences that point to the role each region plays in survival behaviors such as fight or flight. Tonight, Ann will tell us what life has been like since winning the Eppendorf Science Prize for Neurobiology. So, ladies and gentlemen, Ann Kennedy. And Axel, I stayed on script.
Yeah. Oh, yes, I do. You want to back up?
All right. Thank you all so much. Thank you, Axel out there in the cloud and Caroline, for an amazing year and for putting all of this together, and to Science and Eppendorf for making this happen. It's been a fun year. I wanted to show some photos. This is from last year's prize ceremony in San Diego and from visiting Eppendorf headquarters in Hamburg, touring their production facilities, which was amazing, and just learning about some of the things that Eppendorf has supported over the years. And it's been fantastic being able to be part of this network of researchers who have been recipients of this prize in the past. So I want to talk about what we had done and what we've been doing now, following up on the research that was described in my essay last year. I said a lot has happened. The first thing is that the last of the papers that I described in my essay is now published, so that's exciting. And what's cool is it came out in 2023, which is the 50-year anniversary of Lorenz and Tinbergen and von Frisch sharing the Nobel Prize for developing the field of ethology, which is the study of naturalistic, sort of evolutionarily constrained behaviors. And in particular, this work was studying aggressive behavior, which is the species-typical, very stereotyped escalation of different types of behaviors that animals perform to allocate resources and establish territories and mates. And this is a behavior that's been studied across a wide variety of organisms. You find it all over the animal kingdom. Each species has its own species-typical way of showing these behaviors. And there are certain patterns that people found, ethologists found over the years. And one of the things that we were interested in was this escalation of aggressive behavior. And this is what I wrote about in my essay last year. And as much as I like non-traditional model organisms, we couldn't study this in white-tailed deer, but we could study it in laboratory mice. And my experimentalist collaborators in the laboratory of David Anderson at Caltech were performing microendoscopic imaging in hypothalamic nuclei that are involved in the control of survival behaviors including defensive behaviors, reproductive behaviors, and aggression. And the challenge of doing these experiments is you're watching two mice run around, and then you're recording from their neural activity, and you're getting a lot of calcium imaging traces that look kind of like this. And for 6 years of my postdoc, we spent a lot of time squinting at these things and trying to figure out what was going on with these neurons. And the work that I wrote about last year is one of our findings from this, which is that if you do the right bit of dimensionality reduction and fitting of dynamical systems models, you can turn this mess into a signal that has this wild, slowly evolving ramping activity over time. So you introduce an intruder mouse, the animals are interacting, investigating, showing dominance mounting and attack, and there's a signal in hypothalamus, it's a fairly big signal, like 20% of the cells, that gradually ramps up over time. And what we realized after looking at this and exploring this is this gives us a very subtle control knob over the escalation of this aggressive behavior and over how aggressive different individual animals are. And in the work that I write about, we found that these ramping dynamics that we uncovered in hypothalamus had this really interesting relationship with behavior, where changes in the magnitude of this ramping signal were correlated with changes in the intensity of aggression. So the mice start out relaxed, and then you get this gradual escalation of their aggressive motivation, ending in outright attack. But then we also found that if you look at different animals that have different levels of aggressive behavior, the degree of persistent neural activity along this ramp was correlated with their willingness to fight. Some animals get revved up to fight and they're ready to fight and they keep going and this activity stays persistent for a long time. And other animals will get a little bit of increase and then it dies away again really quickly and they never really escalate to outright aggression. So in the year since this was published, we've been doing follow-up work. As a theorist, I'm interested in understanding mechanisms by which neural networks compute things. And so we've been studying this from a theoretical perspective, asking how these subcortical regions are involved in the control of aggressive behavior. And to not bore you with the details, we've been exploring with aggression and with other collaborators, including Nick Betley and Weijia Hong and Moriel Zelikowski, who might be here tonight, the role of neuromodulators and neuropeptides in controlling motivational states, things like aggression, but also things like pain and hunger and stress, and how these neural networks can tune their degree of properties like persistence to alter animals' behavior control policies and grant them this flexible interaction with the world around them. We're also interested just sort of on an algorithmic level. What are the computational principles that underlie this flexible behavior control? How should we be thinking about how the brain is implementing this? And then the other aim of the lab that we've been developing in the year since this project was going towards better understandings of the structure of social behavior itself. So foundation models are in AI topics like ChatGPT and large language models that just try to capture the total structure of a subject domain. And in social behavior, we've been building towards this by collecting large volumes of data from collaborating labs and really performing comparative studies to understand the structure of that behavior. So we're organizing a collaboration to actually do this at scale in 7 different rodent species that have very distinct social structures, all the way from the antisocial blind mole rat to the eusocial sort of insect-like colonies of naked mole rats, and understanding what are the differences between the brains of these species and in the structures of the social groups. We're also collaborating with labs to establish new benchmark data sets for AI and computer vision, pooling hundreds of hours of data from various labs studying the structure of social behavior to really try to tackle this problem at scale and develop computational tools for making sense of how these behaviors are structured so that we can then gain better insight into their control by the brain. But probably the most important thing is over the past year, my lab at Northwestern has grown and it's been a fantastic 3rd year of science. And more than anything else, I'm really grateful for the folks who have come to join the lab and my postdocs, my graduate students, and my staff scientists who've been a part of making this happen, and also for my amazing collaborators who have continued to work with us and help us find other domains where we can apply these computational tools to understanding the neural control of behavior. So with that, I'd like to thank you and thank Eppendorf and Science and you all for the opportunity to talk about my work and present it to diverse audiences. And I'm really excited to pass the torch to this year's recipients so that they can have an amazing year of just getting to know the field and getting to tell people about their work. And so with that, I'm going to hand things off to Peter Stern, who's going to introduce this year's winners and give them a chance to tell about their work.
Good evening, everyone. I'm Peter Stern. I'm one of the editors of Science, and I have also chaired the selection committee for the prize this year and a few years in the past as well. We are in the business of publishing science, and I was starting to think, what is necessary to be a successful scientist these days? You have to be a multitasker. You have to read a hell of a lot. You have to be on top of the discourse in your field. You have to come up with an original new idea, and then you have to perform experiments, and you have to perform them to high standards. You have to do your controls. You have to make sure that your hypothesis is properly tested. So all these parts we probably know, but if you want to be successful, there is something else that comes on top of that. And that is, you have to communicate. You have to tell what you have found, and you have to tell it to your peers in the field, so the other scientists who are working on similar topics. And these days, I think it's getting more and more important. You will also have to communicate with the wider world. You have to tell the others what we are doing and why it is important to support science, and the findings that we have may have implications for the wider world. So when we decided to join forces with Eppendorf to honor someone who is able to communicate, we had exactly that in mind. Young people at an early stage in their career where they bring these things together. So we look at high quality science, but also the ability to write well, to grab the audience, to get your reader to follow through. So, in practical senses, what we are doing is we get all the essays in, and in the first stage, it's like a two-tier system. In the first stage, I hand out the essays to my colleagues at Science Magazine, other editors. Not all of them are neuroscientists, but I can tell you they are all smart. So they have seen tons and tons of texts, and they can read something and they can say, oh, this sounds interesting. This sounds that grabs my attention. I will stay and read it through. So in the first stage, we try to find the ones that are most attractive, where the science is well done, so we weed out all the others. Once we have done this, more or less, it's down to a smaller group of essays, and then we invite outside scientists. For many, many years, we have been collaborating with the Society for Neuroscience. So the society will present us with a selection of jury committees. And there's usually myself, another colleague from science, and then the external judges. I send out just the best of the essays to them, and at some stage we will have a meeting and we will decide which one of them are the best. In the first round, the difficulty is more or less the weeding out. In the second round, it is fun to read because you have already a selection of relatively good essays. The difficult part in this case is to find out who of these good essays is outstanding, who is just a little better, and then we sit together and decide. And over the years, I think most of the time we did really good decisions. I can see some of the past winners. I've seen that they have gone on to further successful careers within the world of science. So I think this is somehow encouraging us to continue the way we've done before. So with this, I think we should start getting our winners this year on stage. And first, we begin with the finalists. The first of them is Michael Skinner. Michael, please come. So, Michael started his career at McMaster University, and then he went to the University of British Columbia, where he did an MD, PhD. And a large part of his PhD experiments were done in Switzerland at EPFL. And I think this is what he is going to talk about today. In the meantime, he has ended up in Princeton University. And the title of his essay was From Single Cells to Neural Circuits. Michael.
So you gotta do this, and you're gonna.
So, our next finalist is Mattia Aime. I hope I pronounced this right now. Mattia started his career in Pavia in Italy, and then he went to Bordeaux in France. And at the moment, he is at the University of Bern. And the title of his essay is To Feel Better, Sleep on It. Doesn't already this title entice you to read the whole thing? That's just an example of how you have to write it, right? So, and now we come to this year's overall winner, Marissa Scavuzzo. Marissa started her career at Baldwin Wallace University in Ohio, and from there she went to do a PhD at Baylor College. And now I find out you've gone back to Ohio. Now you are at Case Western Reserve University. So, and the title of Marissa's essay was The Way You Move. But be careful, she's telling you a lot more about this, and it's probably a little bit kind of misconception. Listen what she's going to say. Okay, so now Michael is first, yeah? Please.
Okay, can everyone hear me? I think it's on, but anyway, I can stand here. So it's really an enormous honor to be here today, and I'm so glad to have the chance to tell you a bit about some of my work on mapping neural circuits in high throughput using single-cell genomics. And this was work that was done during my time as a visiting PhD student in the lab of Grégoire Courtine at the EPFL in Switzerland. And Grégoire, for many years now, has been dedicated to restoring voluntary control of walking after paralysis. In 2012, Grégoire's lab developed a neuroprosthetic device that allowed paralyzed rats to walk again. And in 2018, his lab announced that they had translated this technology into 3 human patients who had been paralyzed after suffering spinal cord injuries. This device that Grégoire's lab developed is essentially a set of electrodes that are surgically implanted onto the dura mater of the spinal cord. And these electrodes sit on the dura and they deliver spatially and temporally precise bursts of electricity to the spinal cord in a way that reproduces the natural activation of motor neuron pools during walking. One of the most incredible outcomes of this work was that after months of intensive neurorehabilitation, all 3 patients regained neurological functions even when the electrodes were turned off. In fact, one patient even regained the ability to walk without any electrical stimulation at all. But the mechanisms that were allowing this to happen were completely unknown. And so the goal of this project that I'd like to tell you a bit about was to discover the neurons that were being engaged and rewired by this epidural electrical stimulation, or EES. But what I think is perhaps an equally important contribution that we made in this study is how we identified this population of neurons. And so I'd like to tell you about the framework that we developed to map neural circuits in high throughput, starting from the beginning. First of all, we knew that we would need to study EES in the lab and not in human patients, and so we would need an animal model of neurorehabilitation. So one of Gregoire's postdocs, Claudia Kath, developed a mouse model that captured the effects of a spinal cord injury, electrical stimulation, rehabilitation, walking, and volition. And Claudia came up with a very sophisticated experimental design with 8 different experimental groups that would allow us to tease apart the effects of each of these factors. Next, since we didn't have a specific hypothesis about which neurons this stimulation was affecting, we reasoned that we would need to somehow measure its effects on every subtype of neuron in the spinal cord at once. And so for this, we turned to single-cell genomics. We used single-nucleus RNA sequencing to measure gene expression in more than 30,000 individual neurons from 36 different subpopulations across 8 experimental groups. And using this data, we could monitor changes in gene expression in every subpopulation of neuron in the spinal cord at once. The only problem was, it wasn't exactly clear what this data was telling us. We didn't really realize it at the time, but we had collected what might have been one of the most complex single-cell experimental designs in existence at this point, and the computational tools to answer the questions that we had just hadn't been developed yet. And so we came to realize that the final missing piece was a way to prioritize the neurons that were being engaged and recruited by EES from this single-cell dataset. Initially, we tried to use markers of neural activation, like the immediate early gene FOS, to identify these perturbed neurons. But these genes turned out to be so lowly expressed in our data that there wasn't really any distinct signal in any one particular subtype of neuron. Then we asked if we could simply count the total number of differentially expressed genes within each neuron subtype, but this again seemed to produce very inconsistent, even contradictory results. And so after months of trying these existing approaches, we realized that there just wasn't an existing tool that would do what we wanted. And so I developed one. The computational method that I developed, which is called Augur, uses machine learning to identify subpopulations of neurons that become transcriptionally separable in response to any given stimulus or perturbation. So what does this mean, transcriptionally separable? The idea is to quantify how accurately we can predict the experimental condition that any given neuron came from based on gene expression alone. So to do this, we pretend that we don't know which experimental condition a subset of the neurons in our dataset came from. For example, we might take 20% of the neurons that we sequenced and pretend that we don't know whether these neurons came from treated or untreated mice. We would then use the other 80% of the neurons in our dataset to train a machine learning model that learns to predict for any given neuron from its gene expression whether that neuron came from a treated or untreated mouse. And we then use that trained model to predict for those other 20% of neurons whether these came from treated or untreated mice. But of course, in reality, we know which mice these neurons came from. And because we know this experimental ground truth, we can quantify how accurately the machine learning model was able to predict whether a given neuron came from an injured or an uninjured mouse, treated or untreated. Intuitively, this will be easy for the model to do for subpopulations of neurons that are transcriptionally distinct between conditions, but it will be difficult or perhaps even impossible for subpopulations of neurons that are transcriptionally similar or even identical. And so the accuracy of this machine learning model provides a single quantitative measure of transcriptional separability that simultaneously takes into account every gene at once. We validated Augur quite extensively in both simulated and published single-cell data, and we showed that Augur was substantially more accurate at identifying perturbed cell types than these existing methods. In simulated single-cell data, we showed that Augur could correctly rank synthetic perturbations of known intensity, whereas in published single-cell data, we found that Augur correctly ranked perturbations of known intensity across species, whereas these methods based on differential expression that we had initially tried to use failed completely. And when we applied Augur to our own single-cell dataset from the injured mouse spinal cord, we found that Augur prioritized the same subpopulation of ventral excitatory interneurons expressing VSX2 in all 6 experimental comparisons. And this result wasn't just robust across experimental comparisons, it was also very robust to the threshold that we use to define transcriptionally distinct neuron subtypes. But to independently confirm this result, we also used spatial transcriptomics to profile the spinal cords of the same mice. We registered 61 mouse spinal cord sections onto the same two-dimensional coordinate system so that we could perform differential analysis of our spatial dataset. And I then extended the logic behind Augur to operate on spatial transcriptomics data in a second method that I named Magellan. Instead of trying to predict what experimental condition a single neuron came from, Magellan is trying to predict what experimental condition a given spatial barcode came from, but the machine learning approach is otherwise very similar. And we showed in a series of simulation experiments that Magellan correctly recovered synthetic spatial perturbations. And when we applied Magellan to our own spatial transcriptomic dataset from the injured mouse spinal cord, we found that Magellan prioritized the intermediate laminas of the spinal cord, which is exactly where the ventral excitatory interneurons that Augur had prioritized were known to be located. This data really pointed to a key role for these neurons in the recovery of walking after paralysis, and that led Claudia and another postdoc in Gregoire's lab, Tom Hudson, to go into the lab and experimentally test their role. And Claudia and Tom showed, using a series of optogenetic and chemogenetic experiments, that these neurons were indeed both necessary and sufficient for the recovery of walking after paralysis. I think that what's so exciting about this work from a technological perspective is that we've shown that with the right computational tools, we can repurpose single-cell transcriptomics into a technology that allows us to identify neurons that are activated by any given stimulus or perturbation. And in fact, we've gone on to use Augur and Magellan in a series of other projects. For example, we recently used Augur to identify a subtype of neuron that could be regrown through an anatomically complete spinal cord injury to restore walking in paralyzed mice without any electrical stimulation at all. We've also used Augur and Magellan to help us decipher an integrated single-cell and spatial atlas of the spinal cord injury lesion site itself, comprising more than 500,000 cells, single-nucleus multi-omics, and 4-dimensional spatial transcriptomics. And ultimately, Augur and Magellan guided us to a new gene therapy approach that we showed can restore the natural recovery of walking that takes place after incomplete paralysis in old mice who otherwise fail to demonstrate this natural recovery. So looking towards the future, I'm optimistic that Augur and Magellan can be powerful tools to help us understand the neurons that are involved in the response to any given stimulus or perturbation, and ultimately to develop new therapies. So with that, I'd like to thank Gregoire as well as my colleagues in his lab who contributed to this work, as well as some of the key funding sources that supported this work, and of course you for your attention.
microphone like Michael did.
Yeah, okay.
Use the hand.
Yeah, I think it's working well, right? Yeah, okay. First of all, I would like to thank Science and Eppendorf for this great opportunity. And of course, my mentor, Professor Adamantiris, and the co-authors of the paper, and some of them are here tonight, and it's really a pleasure. So I would like to present to you my postdoc work at the University of Bern that I presented in this essay under the title, "To Feel Better, Sleep on It." So I'd like to start with this picture. This is my daughter, who is reading a book about emotions. And, well, in this book, this color monster is messing up with his emotions, which are represented by different colors. And this kid is offering herself to help the monster to reorganize the emotions in the correct jar. Well, I'm presenting you that because all my research question is inside this book. During the day, we're bombarded by events and information that are able to elicit different types of emotions. And during the night, sleep is able to reorganize these emotions, and in particular, the information that is linked to these emotions. And in fact, sleep, and particularly REM sleep, has been long-lastingly associated with the processing of emotional information. And this has been suggested by the fact that both in the animal and human brain, neurons that are transitioning to REM sleep, so neurons that are providing the transition to REM sleep in the brainstem, are associated with the activity of neurons that are, or regions that are known to process emotional information. For example, the hippocampus, the amygdala, which is the core of emotional processing in our brains, and also the prefrontal cortex. In fact, the prefrontal cortex has been recently shown to be involved in a restructuring upon an emotional fear conditioning task. And this is strictly linked to the occurrence of REM sleep. In fact, in mice that were REM deprived, not only they found a decrease of the behavioral performances, but also a loss of this reorganization that they observed in the prefrontal cortex. But there is a big paradox in literature, and this is due to the fact that although the prefrontal cortex is important for processing emotional information during REM, it has also been shown to be silent during REM. This has been identified both in human studies and animal studies. For example, these clinical works have found that the dorsolateral prefrontal cortex is silent during REM sleep. And also in animal studies, it has been found that neurons that are activated during REM sleep are much lower in number than the wake-active ones. So when I joined the lab of Antoine in Bern, we wanted to understand how the prefrontal cortex is performing at the microcircuit level during REM sleep. And how can this region store emotional information if apparently it's silent during REM? So in order to do that, I used a technique to photon imaging that allowed me to record a wide number of neurons longitudinally across different states, not only during wakefulness, but then during subsequent non-REM, particularly during REM sleep. And I firstly focused on the pyramidal excitatory neurons, because those neurons, they represent roughly 90% in the prefrontal cortex. I recorded longitudinally during different brain states the cell bodies of those neurons. And in fact, we found that in accordance with the literature, during REM sleep, there is a drop of the activity of those neurons. And you can see also that the frequency of the somatic calcium spikes is indeed decreased during REM sleep compared to the other states. But because those neurons are not only made of their cell bodies, but they also have a wide dendritic arbor, we decided also to record the dendrites of those neurons. And we used the same technique, but in this case, we went more superficial, and we recorded the dendrites of those neurons. And surprisingly, what we found is that both the confined, so small events, but even the events that are spreading across the dendrites, they increase their activity during REM sleep, even compared to wakefulness. So for the first time, we found that there is a decoupling during REM sleep between the activity of the dendrites, which seems to be involved into a disinhibition during REM sleep, while in parallel, the cell bodies of the very same neurons is potentially subjected to an inhibition. And this is exactly what we tried to test right after when we recorded the local inhibitory neurons in the prefrontal cortex. And we firstly recorded the parvalbumin interneurons, which is a population of inhibitory neurons that are known to silence specifically the cell bodies of pyramidal cells. And in fact, what we found is that those parvalbumin interneurons are hyperactivated during REM sleep, and they silence the cell bodies of the pyramidal neurons. In parallel, two other populations of inhibitory neurons, which are the VAP and the somatostatin interneurons, are known to be involved in a process of disinhibition at the level of the dendrites. Those because this first population is inhibiting the somatostatin, which in turn inhibit the dendrites. And what we found is that during REM sleep, the VAP interneurons, again, they become extremely active. As a consequence, they silence the somatostatin interneurons, and this provides a disinhibition at the level of the dendrites. So a reorganization of the local interneurons is sufficient to decouple the activity of the dendrites and the cell bodies of pyramidal neurons. But has this something to do with emotional processing? So in order to test that, we did a classic emotional fear conditioning task, where during the acquisition phase, the mice were subjected to the presentation of two classic auditory cues called the CS plus and the CS minus. The CS plus is positively paired to an adverse event, a food shock, and the animal learns that this tone is predicting for danger, while the CS minus, which is not paired to the food shock, is predicting for safety. And the day after, the animals in our memory retrieval task, they are indeed able to discriminate between the CS plus encoding for danger. In this case, they will respond with a fearful response, which is freezing, while the CS minus is not encoding for any kind of fearful response. But in addition to that, right after the learning, when the animals were back in their home cage, for 4 hours, we silenced the somatodendritic decoupling, so we perturbed this decoupling selectively during REM sleep, or by silencing the VAP interneurons. So in other words, we blocked this dendritic activity, or by silencing the parvalbumin interneurons. So in other words, we reactivated the cell bodies. And this was sufficient to provide two opposite behavioral outcomes. When we blocked the VAP interneurons, the animals the day after, they couldn't discriminate. They were generalizing and responding with a fearful response also to the safe cue. On the contrary, when we blocked the parvalbumin interneurons during REM sleep, the ability of the animals to discriminate was even significantly enhanced compared to their control. You can see that the discrimination score is very poor when we block the VAP interneurons, and is enhanced even compared to the control when we block the parvalbumin interneurons. Finally, we also proved that this mechanism is relying upon local synaptic plasticity, because the brain of the same mice were sliced, and we performed classic companion ratio recordings, which is a clear measure of synaptic plasticity, and found that when we blocked the VAP interneurons during REM sleep, the synaptic plasticity was not occurring because the level of companion ratio was comparable to naive animals, so animals that did undergo the fear conditioning task, and significantly lower compared to the YFP controls, so the controls. On the contrary, when we blocked the parvalbumin interneurons, the synaptic plasticity was even significantly enhanced. So you can see that there is a link between synaptic plasticity and the behavioral output. When we block the VAP interneurons, so the dendrites, no plasticity occurs, and as a consequence, the animal cannot discriminate. On the contrary, when we block the parvalbumin interneurons, we observed increased synaptic plasticity, and this resulted in what we called overgeneralization. And I'm saying overconsolidation because when we tried in a subgroup of those animals to change the rule in the environment and doing a sort of extinction, so now the CS plus is not encoding for danger anymore, those animals in which we block the parvalbumin interneurons are not able to optimize their behavioral response to the changement in the environment. So, to summarize, we found that a somatodendritic decoupling with the silencing of the output, so the cell bodies, and the disinhibition of the dendrites is important for optimizing emotional responses during REM sleep. When we block the dendrites, no consolidation occurred, but when we block the cell bodies, we observe a sort of overconsolidation. So in conclusion, we found a neuronal mechanism that is able to triage emotions during REM sleep, or in other words, we found a mechanism that is able to help this monster to reorganize the emotions in the correct jar. Thank you for your attention.
Can you guys hear me okay? Great. Well, thank you so much to Eppendorf Science for this amazing honor. It still feels really quite surreal, and it does so for many reasons, but one of these reasons is because so many of you guys are hardcore neuroscientists. You guys focus on neurons in the brain. And I not only focus on the non-neuronal cells, I actually study them in the gut. But I hope by the end of this I can convince you guys that the gut is just as interesting and complex as the brain. Before I do that, I'm gonna make you guys look at a picture of my daughter. So just like Maddie, we're proud young parents. So this is my daughter the day she was born. And at this point in time, she wasn't much more than this really cute little blob of flesh. We aren't born knowing how to talk or how to walk or really how to think. These are complex processes that we develop over time. So if you think about the brain in simple terms, it's majorly made up of two cell types, neurons and glia. And at the same time that all of these complex processes are developing, these glial cells are undergoing robust postnatal development. So, as these glia in Lillo's brain developed and matured, she learned how to talk and how to walk. She's now running around and telling stories. And even last week, she told me that she didn't like my shirt. So she can clearly now think on her own. But for a long time, these glial cells in the brain were thought of as nothing other than the glue that holds neurons together. So glia actually translates to the word glue in Greek. But now we know that these cells are super diverse. They do a whole bunch of different things. And if you mess any of them up, it can lead to disease or even death. But this isn't the only nervous system in our body, and this also, thinking, isn't the only complex trait that we develop. So believe it or not, actually babies aren't born knowing how to poop. So if you've ever been around a really young newborn when they're trying to defecate, they get really frustrated and really mad because they just can't figure it out yet. And that's because gut motility is an incredibly complex process, and it requires the coordination of a bunch of different cell types to keep things moving in one direction. Actually, infant dyschezia is the official term for these first few weeks of life when babies, the intestinal muscle just can't coordinate yet. And this coordination is really essential for peristalsis to occur. So peristalsis is the wave-like contractions that happen in our gut that keep things moving in this one direction. So in this video, what you see is the outside layer of the mouse intestine that I dissected and I placed in a dish. So in this tissue, you have pacemaker cells, muscle, neurons, and glia. But this video is not sped up, and this tissue's actually been in the dish for 3 days. There's no brain here. So peristalsis is not controlled by your brain, but rather it's actually another nervous system that weaves through every layer of your gut that's so complex it's often called the second brain. So when we eat food, the food passes by and this force changes the electrical activity deep within the muscle layer, leading to the release of neurotransmitters like acetylcholine from neurons that causes muscle to contract upstream of this food bolus, and neurotransmitters like nitric oxide downstream, causing muscles to relax. And this squeezes things through in one direction. And this process is actually disrupted in a wide range of different diseases and conditions, many of which actually are unexplained in origin, and often gut motility is seen in neurological patients. But neurons don't exist in all of these layers by themselves. And actually, there are glial cells present in every layer of the gut. So from the myenteric plexus down between these muscles and the submucosa and the mucosa, where they have the opportunity to interact with a whole bunch of different cell types. We know enteric glia are not the same as glia in the brain, but the field of enteric glial biology is still in its infancy. And it's my goal to understand how do enteric glia contribute to digestion. But before we can answer this question, we're going to remain in the dark until we can first answer what types of glia even exist in the gut. So to answer this question, we could just shine a light on a couple of these cells, zoom in, look at the cell, and ask it, who are you? Where do you live? What do you look like? And what do you do? So to do this, we could, ask who they are by asking, how do you express yourself? Or what genes do you highly express? But like I mentioned, these cells are weaved through every layer of the gut. So they're in between the really tough opposing muscle layers, they're in the delicate submucosa, and they're up in the villi. So it's actually impossible to dissociate this tissue without biasing for what cell population you're getting. So an alternative that you actually heard from Michael is to just lyse the tissue and isolate these individual nuclei to sequence. This would then allow us, in theory, to profile every single nuclei in the entire gut. So to do this, I developed new technologies that allowed us to sequence nuclei from highly degradative tissue, and then this let us generate a snapshot of all of the cells in the proximal small intestine or the duodenum. So you can see here, just for simplicity, I've labeled them as epithelial and non-epithelial cells. A lot of the intestine's epithelial. And every dot here is an individual nuclei that's transcriptome's been sequenced. What's really nice is we can actually maintain the expected proportions of cell types. So this, for the first time, allows us to capture the diversity of cells in this tissue. So what we can do now is, since we're equipped with this, we can pull out these cells, so these non-epithelial cells, and start to look at what cells are there. And we can capture enteric neurons and enteric glia from all of these different layers of the tissue. If we just focus on enteric glia and enteric neurons, now we can actually cluster them by themselves and get an idea of what the diversity of these cells are like. And we leave neurons in here actually as a control. So here you can see these colors indicate cells that are predicted to be different from one another based on their transcriptome. So it looks like there's a remarkable molecular diversity of glial cells in this tissue. And when we look at the genes that are highly expressed, so you can see here in this heat map, yellow is high, blue is low. On the top you have individual nuclei that are grouped by which cluster they come from, and each row is an individual gene. And what you can appreciate is this nice diagonal yellow line shows that these cells are transcriptionally unique. And we focused for the study on these hub cells, which we named these hub cells for reasons you'll find out soon. So first we wanted to make sure these hub cells were actually a subtype of glia and that they existed in the gut. So we looked at the different transcripts that were enriched in these different subpopulations, and then we used a mouse that let us mark the enteric nervous system in RFP, shown in magenta here. We stained for a marker that's highly expressed in these hub cells called piezo2, shown here. And we not only saw that this protein's expressed, and that it's only expressed in a subset of enteric nervous system cells, so this green piezo2 positive cell, it's not overlapping with all of these magenta cells. But one thing that really stood out is that they were entirely restricted to the myenteric plexus. So they only existed here between these two opposing muscle layers. We then wanted to know, what do they look like? So we used sparse in vivo labeling to look at these cells, coupled with 2-photon microscopy, and we're able to see that these cells were almost entirely the same morphology. So now we can start to answer some of these questions. We see that enteric glia are diverse, and hub cells are one of these many subtypes. We see that these cells express different proteins, like piezo2, and that they're entirely restricted to the myenteric plexus, where they're close by to pacemaker cells, neurons, and muscle. We also see that they're almost entirely interganglionic in morphology. But what do they do? So since we're uniquely equipped with these, this global transcriptional map of the small intestine, we can now do really high-quality interactome analyses where we can look to see and predict what these different cell types interact with by matching different expressed ligands and receptors. And by doing this, we're able to see that these hub cells are predicted to interact with pacemaker cells and neurons, two cell types that we know they live right next to. When we look at functional analysis, we see that they're enriched for neuronal action potentials. When we look at what genes are involved in neuronal action potentials, we see again piezo2 popping up. So this transcript keeps showing up. What is piezo2? So piezo2 is a mechanosensory ion channel that sits in the membrane of your cell. So whenever force is applied, this channel physically opens and it allows ions like calcium to rush into the cell, causing it to become activated. So it's actually how we can feel touch. It's how we can feel hugs. So in this image, this is my husband, Andrew, who's sitting over there, hugging our daughter after she woke up from a nap. And they can actually feel this hug because of piezo2 is expressed in the Merkel cells in their skin. It's also expressed, if everybody takes a deep breath with me, it's in the neurons that innervate your lungs, so you can feel your lungs stretch when you take a deep breath because of that. And recently, beautiful work by Rocio Servin showed that it's actually how we can feel fecal matter in our distal colon. So we see that piezo2 is expressed in these glial cells, in these hub cells. We see it's expressed at the protein level. So here in green, showing these glial cells, the nuclei marked in white, and we see piezo2 at the protein level. So does this mean that these cells can sense force? So to test this, we developed a new device that allowed us to measure the activity of glial cells while applying force. So I carefully dissected off this outside layer of the tissue where we know these hub cells live, and I placed it into these modified intestine-on-a-chip devices. Then we can hook up to microfluidic pumps to apply shear stress force at a specific dyne to stimulate the activity of these piezo2 channels. And all of this, while we're doing it, we're doing high-resolution calcium imaging. So here in this video, what you're gonna see is force applied. So you're gonna see a flash of light, that's actually the fluid moving by. And then in these two yellow circles, you're going to see cells that are active. And there's a still image of this here, and quantification to the right. And what was really interesting is we saw a subset of these cells were mechanosensitive, so they were not active at baseline, but when we applied force, they were active. And most of these cells that were mechanosensitive, when we applied an inhibitor to piezo2 to the culture, this activity was actually reversed. So this supports that these cells actually are able to sense force and that piezo2 opens, allowing calcium to rush in after force is applied. We still don't know what they do, right? So we made a mouse that allowed us to delete piezo2 from all adult enteric glia. So on the top here, you can see enteric glia in the myenteric plexus in this muscle region are marked their nuclei in white, and GFAP marks a subset of these cells here. Piezo2 is normally expressed, as I said, in a subset of these cells. But using this mouse line, these cells still are present, but we delete the protein, so it's no longer there. And there's transcriptional quantification to the right. So then we took these mice that have piezo2 deleted from enteric glia, and we force-fed them a red dye. Then we put a timer on and just waited until they pooped. And this is what it looks like. We're looking for this red poop that appears. And here's quantification. When we force-feed these mice this red dye, when the mice that no longer have piezo2, there's an increase in the time it takes for them to pass this red dye. So there's an increase in transit time. And every dot for all of these plots I'll show you are in individual mice. We next wanted to know more about how this was happening and where things were getting stuck. So we fed mice a fluorescent molecule, and then after 30 minutes, we looked throughout the gut to see where it was distributed. So here, every well is an equally sized segment of the intestine from the beginning to the end. And these normal mice, most of the dye you can see is here. But when we delete piezo2 from enteric glia, everything shifts earlier. And that's quantified here. So you can see, again, this is showing further here in these mice with piezo2, and when you delete it, everything's earlier in the gut, and things actually are building up in the stomach. So this is a measurement of how much dye is stuck in the stomach. So does this mean that there's defects in gut contractions or gut motility? So we went back to our intestine on a chip devices, and we applied force and then measured their contractions. So here in a normal mouse, you can see this nice rhythmic oscillations that occur. But when we delete piezo2, the amplitude or strength of these contractions is not significantly different, but there's a huge change in the frequency of these contractions. We know nitric oxide and acetylcholine are two neurotransmitters really important for this process, like I mentioned at the beginning. So we looked in the myenteric plexus and measured the levels of these neurotransmitters, and we saw remarkable disorganization in neurotransmission if you delete piezo2. So now we could say that enteric glia can regulate gastric emptying and peristalsis through these mechanosensory ion channels piezo2. But so far we can't tell you if this is a subtype of cell doing this because we deleted it from all enteric glia. So this is what I had just shown you. So when we delete piezo2, there's everything gets stuck earlier in the intestine, and there's a buildup of contents in the stomach. We know enteric glia are not the same as glia in the brain, and they actually express canonical glia markers in really weird combinations. So here, looking throughout the entire gut, these are different subpopulations of cells that were predicted. You can see most of the cells that express piezo2 co-express GFAP, while those that express PLP1 don't have as high of expression. So we use these different genetic markers as tools to drive the deletion of piezo2 in different populations of enteric glial cells. So when we delete piezo2 from GFAP positive cells, we see things get slowed down and the dye builds up in the stomach. So we again see a phenocopy where there's a defect in peristalsis and gastric emptying. But when we delete it from PLP1, there's actually not a difference. And if anything, it's trending the opposite direction. So this shows for the first time that these enteric glial cells are functionally specialized to do different things. So now we can actually start to answer this question. What types of glia are in the gut? There are functionally specialized subtypes. So we see enteric glia are diverse, and hub cells are one of these cell types. We see that hub cells are almost entirely interganglionic in morphology, and that they live in the myenteric plexus nearby to pacemaker cells and neurons, where they have the opportunity to modulate electrical activity. These hub cells express the mechanosensory ion channel piezo2, and when force is applied, this channel physically opens, and it allows these calcium ions to rush into the cell, causing the cell to become activated. So these hub cells actually act as signaling hubs, where they can integrate this information and then spread it to nearby cells. This causes these other cells to become activated in the regulated release of different neurotransmitters like acetylcholine and nitric oxide, and thus regulation of peristalsis. And ultimately, this leads to changes in whole animal physiology because it regulates and impacts gastric emptying and gut motility. And this is also really interesting because it means that these hub cells could actually be contributing to the many different disorders and diseases that we know gut motility is affected in. And in fact, actually, the phenotypes I showed you strongly resembled gastroparesis, which is a defect in gastric emptying. This is a condition that's very poorly characterized and unusually common in neurological diseases. And actually, patients with Parkinson's disease, gastroparesis is actually usually seen before the onset of neurological symptoms. We also found that in aging, piezo2 expression decreases in the myenteric plexus. And we know that there's widespread gut motility issues in aging. So this indicates that perhaps these cells or their function is changing in age. But overall, this is a major conceptual shift, because if there are different functionally specialized subtypes of enteric glia, that means that these different subtypes could be functioning differently or dysfunctioning in a wide range of different diseases. And actually targeting these different subtypes could actually provide therapeutic insight for not only the millions of gastrointestinal GI patients, but also neurodiverse patients that experience gut disturbances. And this overall will allow us to understand and reveal more about how digestion occurs. So I really believe we're in the midst of an enteric glial cell renaissance. We know enteric glia are diverse, but now we know they're functionally specialized. And hub cells are one of these cell types that we've seen. It fine-tunes gastric emptying and peristalsis. And this is just one cell type and one function we've defined. Hub cells probably do a lot of other things that we still have yet to figure out. And this really is the tip of the iceberg. There's many other subtypes that we still have yet to functionally define that are different in all of the different regions of the gut. These enteric glia are everywhere from your esophagus to your rectum. And we have to now start considering these different subtypes and how they might be changing in many different conditions. So with that, I want to thank especially Eppendorf Science and the team of researchers that have helped with this project, especially Paul Tizar, my mentor, who is here. You guys should meet him. He's amazing. And my husband, Andrew, who is phenomenal, and our beautiful daughter, Lilo, learning about the brain at an early age. And she actually helped with some of this data. Here she is annotating some of these epithelial cells. So get them started young, right? Thank you.
Thank you. Use the microphone. Thank you, Marissa. That was wonderful for such a great insight into your work. I'm sure you are of my opinion when I say the talks today were amazing, from Mattia and Michael as well, and also from Anne. Thank you very much. I would just like, in closing this evening, to thank once again the prize committee, selection committee, AAAS for the use of their building, thank you. And also all those involved in the organization of the prize ceremony. So we have Amy, Zena, Axel, and Roger, you know who you are. And my thanks also go to you, our audience, for attending today. And I hope to see you all in October, on October the 6th, 2024, at the next prize ceremony when we are in Chicago. Now I'd like to invite you to enjoy a flying buffet dinner. Why a flying buffet? Well, the food comes to you, you don't go to the food. The food itself is from one of the leading caterers in Washington, Elizabeth Petty. So I would say enjoy your evening with good food and inspiring conversations. Thank you very much.